In the previous session, we discussed about the experiments to prove that DNA has the potential genetic material. Today, we'll be discussing about nucleic acid, the structural elucidation of DNA and its properties. So, nucleic acids are one of the vital macromolecules in living system. These are polymers made of monomeric units called nucleotide. It was first discovered by Friedrich Miescher, isolated from nuclei of white blood cells. It was initially named nuclein as it was isolated from nuclei. There are two major types of nucleic acid, the deoxyribose nucleic acid and the ribose nucleic acid, which are generally termed DNA and RNA. The function of DNA is widely known for contributing to inheritance, while the major function of RNA is protein synthesis. We will study RNA in detail in the further classes. DNA is present in the nucleus as a compact structure in the form of the chromosome. There is a detailed structural condensation of DNA around histone protein known as nucleosome. They function primarily in storage and transmission of genetic information, but they may also have structural and catalytic roles. Nucleoside is the sugar plus nitrogenous base, and nucleotide is the nucleoside plus phosphate. This is the picture of the nucleotide and nucleoside present in both DNA and RNA. The major difference in DNA and RNA is the thymidine and uracil bases. We find thymidine in DNA, whereas uracil in RNA. The nucleotides here are deoxyadenylate, deoxyguanylate, deoxythymidylate, deoxycytidylate. And in RNA, instead of deoxythymidylate, we find uridylate. The purines we find are adenine, that is 6-aminopurine, guanine, 2-amino-6-oxypurine. Pyrimidines are thymine, 2,4-dioxy-5-methylpyrimidine, cytosine, 2-oxy-4-aminopyrimidine, uracil, 2,4-dioxypyrimidine. As mentioned earlier, this is written down here. Then coming to the structure of nucleic acid, that is the double helical structure given by Watson and Crick. It was postulated by James Watson and Frederick Crick in the year 1953. It is a right-handed double helix bound around an arbitrary axis. The hydrophobic backbone of alternating deoxyribose and phosphate group are on the outside of the helix facing the surrounding water. The offset pairing of each strand creates a major groove of 22 angstrom and a minor groove of 12 angstroms on the surface of the duplex. The major groove is rich in chemical information. The edge of an AT-BC base pair displays chemical group hydrogen bond acceptor and hydrogen bond donor. And the minor groove has less chemical information. They have complementary sequences on the two strands wound in anti-parallel orientation held together by hydrogen bond and base stacking interactions. The base pairing is on the opposite strand where adenine pairs with thymine with double bond, whereas guanine base pairs with only cytosine with triple bond. This is known as Chergov's rule of base pairing, as adenine is always base paired with thymine and guanine always pairs with cytosine, and they are usually found in the equal amount. That is, always adenine is equal to thymine and guanine is always equal to cytosine. And it has 10 base pairs per turn. One complete turn is 36 angstrom and the width of this is 20 angstroms. The hydrogen bond will help in the thermodynamic stability of the helix and specific of base pairing, when strands come together in double helix, the water molecules are displaced from the bases. This creates disorder and increases enthalpy, thereby stabilizing the DNA. The stacking interaction is the electron cloud interaction, that is pi-pi interaction, helps in stability. The base stackings are attached to each other by transient induced dipole between the electron cloud phenomenon called van der Waals interaction. This is the hydrophobic effect. The ability to form hybrids between two single-stranded nucleic acids is called hybridization. This is the picture of a DNA polynucleotide chain showing 3'5' phosphodiester linkage that connects the 3' carbon of one nucleotide to the 5' carbon of the next nucleotide. So coming to the features of DNA, it is composed of two polynucleotide chain, like mentioned earlier, twisted around each other to form an helix. Hydrogen bond help in the thermodynamic stability of the helix and specificity of base pairing when strands come together in double helix. Hyperchromicity is the DNA absorbs UV light at 260 nanometer. When the temperature of the solution is raised to boiling point of water, the optical density remarkably increases. This phenomenon is known as hyperchromicity. This is due to that the duplex DNA absorbs less UV light, 40%, than the single-stranded chain. It is due to base stacking, which diminishes the capacity of bases in duplex DNA to absorb UV light. At high ionic strength, the negative charge on the phosphoryl group are shielded by cations and thereby stabilizing the helix. This picture on the left shows the denaturation and renaturation of the double helix. DNA strand can denature, that is, the double strand comes apart to when heated above 100 degrees Celsius or at conditions of high pH, and this is known as denaturation. You can see in the picture this is double-stranded DNA, and when heated above 100 degrees Celsius or at high pH, it unwinds itself to form a single strand. This is a temporary or a reversible process. When the solution regains its original temperature and pH, it comes back to normal double-stranded structure, and this is known as renaturation. And the melting temperature at which this characteristic of DNA is largely determined is the melting temperature, and it is usually determined by the ratio of GC content of DNA and ionic strength of solution. That is, higher when the GC content is higher, the melting point is higher. Higher the salt concentration, higher will be the temperature. The melting point is given when the graph of optical density of DNA against temperature is plotted. There is an increase in absorption abruptly over relatively narrow temperature range. This midpoint in transition is known as melting temperature or Tm. Thank you. If you like the video, please like, share and subscribe to our channel.